So we have our last lecture now on our lipid metabolism chapter, and we have two main things to cover in this lecture. We're going to talk about regulation of fat metabolism, and then we're going to talk about cholesterol biosynthesis. And related to that, we're going to talk about not only the biosynthetic pathway, and talk about some kind of gene regulation aspects of that, which tie into the receptor-mediated endocytosis that we talked about in our first lecture. But we're also going to talk about kind of some cellular receptor components to it, and thinking about how some drugs can help affect cholesterol levels. So before that, just again, a quick review of, we've talked about two main kind of metabolic fuels. We've talked about carbohydrates and we've talked about lipids. We'll get to proteins in our next chapter. But again, primary metabolic fuels include glucose, and again, we obtain that via carbohydrate metabolism and then the breakdown of endogenous glycogen. In this chapter, we've talked about fatty acids, again, derived from triglycerols. Again, we have both endogenous and exogenous sources of those. But it's important to remember and distinguish between hydrophilic circulating fuels and those that are hydrophobic. So hydrophobic circulating fuels include our triglycerides that are going to be found in dietary source, again packaged as chylomicrons as they exit the intestinal tract, and then VLDL particles. So endogenous triglycerides that are made in the liver are then circulated to the peripheral tissues via VLDL particles. But both of these particles are essentially the same low density, full of fats, and again circulating to peripheral tissues. We also talked about free fatty acids being complexed with albumin and circulating in the bloodstream in that way. But again, hydrophilic circulating fuels are obviously the most important ones that we maintain certain levels of. So glucose, again, maintained at 5 millimolar. We talked a little bit about lactate, again, lactate being exported as a dead-end metabolite. We'll talk more about that in a couple of chapters in terms of its reconversion to glucose via the Cori cycle. Ketone bodies, again, a way of taking a hydrophilic fuel like triglycerides and converting it into a hydrophilic fuel like ketone bodies. And again, we'll get to protein metabolism and amino acids in our next chapter. So to kind of summarize the components of fat metabolism that we've talked about, we spent a lecture talking about fatty acid oxidation. So that beta oxidation process occurs in the mitochondria, converts fats to acetyl-CoA, and depending on the needs of the cells, either converts it to citric acid or allows it to go to the citric acid cycle, or converts it into ketone bodies. Again, ketone bodies being able to be an exported form of that fuel. Last lecture, we talked about fatty acid biosynthesis. So again, that's a cytosolic process that allows us to synthesize fatty acids from excess acetyl-CoA, and then that activated acetyl-CoA when we've carboxylated it as malonyl-CoA. Again, that occurs in the cytosol. So again, last part is to kind of think about how we regulate these processes. So again, fats will be stored when we have excess energy. Fats will be mobilized when we have a need for energy. So kind of the interplay between these is going to be really important to consider, just like we talked about with glycogen. I'm not going to mention really anything about this figure. This is one that we'll dissect and spend a lot of time kind of breaking down in our activity. So again, an important figure that allows us to not only think about compartmentalization here, so fatty acid biosynthesis occurring in the cytosol, fatty acid oxidation occurring in the mitochondrion of hepatocytes, so being processes that occur in the liver, but we'll also tie this into processes that are going to occur in distal tissues like our adipose tissue. So again, regulation is going to focus on that acetyl-CoA carboxylase enzyme, that ACC enzyme. So we'll kind of walk through this figure thinking about both a fed state and a need state. We'll think about the hormonal signals. We'll not only think about how this affects fat metabolism, but we'll also kind of reflect on how it regulates carbohydrate metabolism. So again, we'll deal with this in both a fed state as well as kind of a need state. So again, we'll reserve those details for our activity tomorrow. Again, the second main topic in this lecture really deals with cholesterol. And so we're going to talk about not only the biosynthetic pathway of cholesterol, so we'll learn some details with that, and kind of what's a pretty interesting pathway to essentially take acetyl-CoA derived carbon atoms and convert them into a fairly complex steroid molecule. But we'll also talk about how this is affected on a gene regulation level. Prior to now, we've really only talked about sort of short-term regulation pathways with allosteric regulation and then covalent modification via phosphorylation events. And we still will talk about those with fat metabolism. But with cholesterol biosynthesis, we're going to talk about gene regulation. And so it'll be an opportunity to kind of review some aspects of gene regulation. We'll also then talk about this from a big picture in terms of thinking about circulating HDL and LDL levels and some disease states that relate with those. So I'm not going to say too much about the details of cholesterol biosynthesis other than to point out the fact that HMG-CoA is an important precursor for cholesterol biosynthesis. And so we talked about how HMG-CoA is synthesized. We make it from acetyl-CoA and it's part of the ketone body pathway. So once you make HMG-CoA, we're able to then use that to make a 5-carbon piece here that we have in this isopentenyl pyrophosphate. So that's going to become a really important thing to think about, is that we've got these 5-carbon pieces that we're going to zip together to make a 30-carbon cholesterol. So math probably isn't too hard to think about. We're going to need to have 6 of these 5-carbon pieces to zip together here. And it's kind of some interesting chemistry that we have there. We'll deal with the process of this in our activity tomorrow. So we've got this isopentenyl pyrophosphate and then this dimethylallyl pyrophosphate. These are just isomers of one another. We have enzymes that interconvert them. We've got these 2 5-carbon pieces that come together to make a 10-carbon piece. We add another 5-carbon piece and then we generate a 15-carbon piece. 2 of these 15-carbon pieces come together head to head, and we have our 30-carbon precursor, which we call squalene. And if you look at this molecule, it looks nothing like cholesterol. So again, we're going to have this molecule that we zip together and functionalize to make cholesterol. And that's kind of an interesting story here. Basically, 2 steps to take this afunctional linear squalene molecule and make something called lanosterol that looks very similar to cholesterol. So pretty interesting biochemistry that happens in 2 steps via 1 enzyme to give us this cholesterol precursor. What's kind of interesting now is that we have 19 more steps to basically do a little bit of functionalization here to interconvert lanosterol and cholesterol. We're not even touching that from a metabolic standpoint, but we will spend some time, like I said, talking about how we regulate this biosynthetic pathway. This looks really busy, and I know there's a lot of writing on here now that we will get to in our activity, but we're going to spend some time, since we don't do really a lot with gene regulation in this class, talking about how we regulate cholesterol biosynthesis. So this will be, again, another figure that we will pick apart in some gory detail in our activity in class. So again, cholesterol gets a bad rap. People talk about good cholesterol, bad cholesterol, and remember, cholesterol is that 30-carbon molecule that we just showed you, or derived from that 30-carbon lanosterol precursor. So cholesterol has good and bad uses. It has good and bad transporters. So cholesterol is what it is. And again, there's lots of good uses for cholesterol. Our steroids are derived from it. We make bile acids from steroids, and the main way of disposing of cholesterol is converting it into bile acids that we can then degrade in our intestinal tract. And so lots of good aspects to cholesterol. Your body needs cholesterol. You can't survive without cholesterol. But again, high circulating levels of cholesterol are where it's problematic. So we call this hypercholesterolemia. That's kind of the medical terminology for high circulating cholesterol. And again, it's important to distinguish whether it's cholesterol that's part of HDL particles or LDL particles. So again, atherosclerosis, we'll talk a little bit more about the pathophysiology of this tomorrow, thinking about what happens when you have excessive circulating cholesterol and why that's problematic from a cardiovascular standpoint. So we'll save those details for tomorrow. And again, this is another figure that we will pick apart in gory detail tomorrow to talk about kind of what happens with cholesterol transport in a normal cell. And what happens in 2 kind of genetic diseases where you have something called familial hypercholesterolemia, and then a specific situation where you have something called Tangier's disease. So we'll break about both of those diseases, as well as understand the normal kind of progression of events in our class activity. Kind of related to that, we'll talk about cholesterol lowering drugs. And so we'll talk about some common drugs that are on the market, and we'll kind of think about how they relate to either the biosynthetic pathway, cholesterol transport issues, and we'll understand how these drugs kind of come into play. And that summarizes and wraps up the chapter on lipid metabolism.